In this video, we are talking about the larval forms present in Fasciola hepatica. So students, in the previous lecture, we have discussed the life cycle of Fasciola hepatica, and where we have discussed the different types of the larval forms which are present in the life cycle of Fasciola. So here in this particular lecture, I will be emphasizing on the structural you can say aspects of these larval forms. Now, first of all, the larval forms in Fasciola hepatica are: number 1, it is a miracidium; number 2, it is a sporocyst; number 3, it is the radia larva; number 4, it is the Cercaria larva; and number 5th, it is a metacercaria larva. So students, the very first form is the miracidium larva, which is hatching out, which is coming out of the capsule whenever the capsule it reaches the water body. Now this capsule, it has got a cap, and whenever the cap it opens, the first larval form or the miracidium it comes out. Now if we consider the structure of one miracidium, it is just like this. So students, I have told you in the very previous lecture that miracidium has got 4 major things which are to be considered very important in case of this particular larva. The first thing is the presence of the eye spots. So these are the 2 eye spots which are present in miracidium, with which it can find out the limnia snail. So this is the larval stage which is founding the limnia snail. So these were the eye spots. Number 2, it has got a brain. So here it has labeled as a brain. So this is the second most important thing which is present in this particular larval form. Number 3, it is the presence of the penetration gland, and here is the presence of the apical gland. So these glands, they are helping in the penetration of this miracidium larva into the snail. Next thing which is very important is the presence of the cilia. So these are present all over the body of this larval form, and they are helping in the swimming of this particular miracidium. So students, these are the major important things in this particular larval form. This is the very first form which is called as the miracidium larva. Now if we consider the structure or the, you can say, the development of this particular larval form, now it is hatching by a hatching enzyme. Now this particular enzyme is opening up the cap of the capsule. Now this particular larva, it excretes out some hatching enzyme which is dissolving the hard shell of the snail. So this is a very, very important thing. Now this particular larva, it is having 21 epidermal plates. Now these plates, they are seen in 5 rows. Now the first and the second row has got 6 and 6 epidermal plates. The third has got the 3, fourth row has got 4, and the fifth row has got 2 epidermal plates. So this particular larva has got an overall of 21 epidermal plates. So if we talk about the eye spots, these are the concavities which are facing away from each other, and they have got a refractile material which is serving as a lens in these eye spots. So if we go with the functioning of these eye spots, these are actually photoreceptors. So if we go with the physiology, this particular larva, it does not feed. They try to penetrate some other animal. So that particular animal is the Limnia snail. So those Miracidia will be surviving which are finding the Limnia snail. So whenever it finds the Limnia, it penetrates the snail and it goes to the digestive glands of Limnia. So in 14 days, this larval form, it is developing into the second form, which is sporocyst. So it takes 2 weeks to form sporocyst. So here you can see the presence of the germ balls inside the miracidium. So these germ balls are giving rise to the next forms, which are the sporocyst larvae. Now students, this is the second larval form, which is the sporocyst. So this is about 1 mm in length, and here there are no cilia present. So whenever the miracidium is penetrating a snail, the cilia are lost. So this second form, it is not having any kind of cilia. But here the cilia, they are replaced by the outermost layer, which is the cuticle. So here it is having a cuticle over it. Now it moves inside the snail and it is getting nourishment from the snail, and the germ balls here which are present, they are referred to as the germ cells over here, and they are forming the next larval form inside it, which is the radia larva. So you can very well see we have written oldest radia. That means this is the next larval stage which is formed inside the body of the sporocyst. So this is a sporocyst larva which has got a radia larvae inside it. So what happens? A single sporocyst larva can give rise to 5 to 8 radia larvae. Now we are going for the third larval form. So this is one radia larva which is coming out of the sporocyst, and this radia larva is coming out by the rupturing of the sporocyst wall. So it is about 1.5 millimeter long, and it is also present inside the snail. It has got a definite mouth, as you can very well see on the top. So it has got a mouth, and this is a very, very peculiar feature of this larva, the presence of the collar. So this is a hard structure which is present near the mouth, and this is called as collar. And the second very important thing in this larva is the lappets. So here you can see the presence of two lappets which is present on left and right side of the radia larva. So this is a single lappet. So these lappets, they are helping in the movement of this larva inside the snail. Now this larva has got pharyngeal glands. So here on the top you can see the pharyngeal glands. So it can feed to some extent. So all the flame cells, they open out by same excretory pore. So this is a very, very important thing, and it is very close to a fasciola hepatica. So it has got protonephridia. So here you can see the presence of the protonephridia. And again it has got the germ cells. So these are the germ cells. And what the germ cells are forming, they are forming the next larval stage, which is the cercaria larva. So here you can see a single cercaria. This is another cercaria which is present inside this radia larva. So they are making the next form, the cercaria larvae. Now here this is very important fact to tell you, is that during the summertime, this radia larva will form more radii. They will not be forming cercaria. So in summers, radia, they form radii, many radii. In winters, this radial larva will form cercaria, the next larval stage. So that means to complete the life cycle, it has to form cercaria. And the cercaria, they are only and only formed in the winter seasons. So that means during winter, these radia will form the next larval stage, the cercaria. So this is very important feature. And there is a definite birth pore which is present over here. And these cercaria larvae, they are coming out of this birth pore. So that means there is a definite birth pore. There will be no busting out of the membrane. So that was about the radia larvae. Now we have the next larval stage, which is the cercaria larva. So one radia, it can form 14 to 20 cercaria larvae. So they come out of the birth pore of the radia. They resemble an adult. So here you can very well see that if you take the body part of this cercaria, this is very comparable to a fasciola hepatica. So here we have the acetabulum. So here you can very well see this is the acetabulum. It is again present in fasciola hepatica. You have the flame cells. Again, they are present in adult fasciola hepatica. It has got a mouth, very comparable to the adult. We have the oral sucker, very comparable. The same pharynx. You have got the esophagus. You have got the intestine limbs. So this whole body is very comparable to the adult fasciola hepatica. Now, this particular form or the cercaria larva, it has got a cystogenous gland. Now, this gland, it secretes the cyst wall around this particular larva. It can secrete a cyst wall. So, as this cercaria is making its next form, the metacercaria, by forming a cyst wall, so this cystogenous gland, it is helping in the formation of the cyst wall. Now here, the presence of the germ cells, they are representing the rudiments of the adult genital systems. So that means these germ cells will be forming the various organ systems in the adult fasciola hepatica. So here you have the excretory duct, which is again very resembling to the adult fasciola hepatica. So students, that was the cercaria larva, and it is coming out of the snail because it is having a tail. So by the lashing movement of the tail, it will swim out of, first of all, it is coming out of the snail, then it will swim, it will coming out of the pond, it will stick to some leaves, and then with the help of the cystogenous glands, it will be secreting the cyst wall around it. The tail will be lost in the water, and only the body, only the upper part of the body, this much part of the body will be under the cyst wall, and it will be forming the next larval stage, which is called as the metacercaria. And this metacercaria is just like this in structure. So here you can very well see the presence of the cyst wall. So this is the cyst wall which is present in this metacercaria. And we have the mouth, we have the oral sucker, we have got a pharynx, we have esophagus, we have got the intestine, the excretory canals, we have got the flame cells. So this is altogether very close to the adult fluke. And now this metacercaria larva is present on the leaves, and this larva is also called as a juvenile fluke. Juvenile means small. So it is reproductively immature, but it is very comparable to the adult. So this is also called as Merita, Marita, Merita. So this metacercaria larva can remain on the leaves for many of the days, many of the months, and whenever a sheep is eating this metacercaria, the cyst wall will be broken down and a fluke will develop out of this larval form. So students, that was all about the larval forms in Fasciola hepatica, and I think that you have understood the structures of these uh, larval forms and what is the life cycle of the whole Fasciola hepatica. So this is a very important question as per your examinations are concerned. So you should be taking a good care of this question. Thank you very much.